Now, there is another very interesting application of antibody-antigen reaction, and that is in the analytical field to develop the biosensors which can be used for analytical or for the diagnostic purposes. Now, what are biosensors? Biosensor is a device which is composed of a biological element and a physicochemical transduction part, which means that there is a biological element. This biological element, this determines the signal, this takes the signal, and further there are physicochemical methods which transduces it. Transduction means enlarges it, enhances it, so that we get the physical indication. Now, nature has given us already very, very precise biosensors like vision, hearing, sense of touching, and things like this. And these are very, very precise methods. You see, our eye, they are the camera which even the present modern technology is still far behind to develop such a biosensor or such a vision development. Now, let us see that there are different biological components which we can use to develop a biosensor. Like enzymes come first. We know enzymes are biocatalysts. Enzymes can perform chemical reaction very fast. And the enzymes, they are used to catalyze a chemical reaction. And then which generates an electrical charge on an electrode, and that can be further transduced or read by a component. Another part is the antibodies. We know that antibodies, antigen, they have a potential to the excellent biological molecules to be used for this technology. Because antibodies, they are very specific. Antibodies, they are very sensitive. Antibodies, their response is very quick. So whenever we have to develop a biosensor or an analytical device, we have to keep in mind these points, that it has to be precise, it has to be quick, and it has to be a constant. And antibodies also stand or become a candidate, best candidate for the biosensors. Now, antibodies may be bound onto thin layers of gold, which in turn are coated onto the refractive glass slides. Antibodies can be bound, immobilized onto the thin layer of gold particles, which can be then bound or coated into the refractive glass slides to make an electrode. And then we take use of another phenomena, which is a surface plasmon resonance. This is called surface plasmon resonance. It means that when the slides, those glass slides on which we have immobilized the antibodies, they are illuminated at a precise angle with fixed wavelength of laser light, then electron waves are generated. Electron waves are produced on the surface of the gold, which is coated with that antibody. And if the antibody it binds antigen. If it is binding with that antigen, then the surface plasmon resonance pattern is changed. This is a very, very precise change and accurate one. So, in that case, the surface plasmon reaction is changed when it is illuminated with a light or with another rays. Now, another important part is a fiber optic sensor. So, you see, we have got now different types of, we are discussing the biosensor. First, we discussed the enzyme biosensor, then the antibody biosensor, and now is the fiber optic biosensors. Now, these sensors, they have been developed, which rely on the natural ability of biological material to fluorescence with the light at defined frequency. So there are natural biological compounds, when they are lighted up or when they are studied under the certain material fluorescent light, they give a certain frequency, and that can be determined or that can be judged from the fiber optic there and gone further for analysis. The reaction vessel then is coated with again antibody and fiber optic sensors used to illuminate and read the light which is scattered from the vessel, and it is further enhanced or further read by a computer or other methods which are there. The sample is then applied to the sample vessel. It is washed, that is the sample vessel which has been coated with the antibody. The fiber optic sensor is again used to illuminate and read the background scattered from the vessel. That gives us the light or the zero reading. And the changes in the fluorescence will give a change in the observed return light, and from that we can determine the concentration of the biological compound or a biological material which is present there. Another biosensor is a piezoelectric crystals. Now, the piezoelectric crystals are those crystals which generate characteristic signature resonance when they are stimulated with an alternating current. That is, when alternating current is passed or there, they generate a characteristic sound, characteristic signature of a resonance, and that can be read. These crystals, they are elastic and change to their surface, and they will produce a change into the signature resonance. So it means the factor which alternate the signature of the piezoelectric biosensor is that the binding antigen to antibody, when they are located on the surface of the crystal, they will produce the different type of the resonance signature which can be read from the piezoelectric crystal, the signal which is produced, and again those signals are extremely sensitive. So we see that the different biosensors which can be devised, which are devised and frequently used in the analytical methodology of an advanced molecular biology method.